Hello students, welcome back to the video solutions of your FLT, that is full length test 1. I am Dr. Ronia, your botany faculty at Memoneet. Okay, so in this video, we are going to discuss in detail about each and every question of your botany starting from 101. And we are going to discuss why is the correct answer and why is the wrong answer wrong. And I hope all of your preparation is going very fast. If it is not going fastly, then start. Even if it is slow, it's good. At least you are preparing. And if you are not preparing, then start because jab jago tab savera, right? So let us dive into the questions. So what is first question saying? The first question is saying the four haploid nuclei formed at the end of meiosis differ from one another in the genetic composition. This difference in genetic composition of the four haploid nuclei that are formed at the end of meiosis is due to what? Okay. So if you all know that meiosis is unique. Meiosis is unique. Now let us first know what is meiosis. So meiosis is a type of cell division. Let's suppose we have a diploid cell in our hand. Let's suppose we have a diploid cell in our hand. This diploid cell is now undergoing meiosis 1 first. After meiosis 1, this diploid cell will form two haploid cells, and these two haploid cells again will undergo meiosis 2 to form four haploid cells. Okay, to form four haploid cells. Now, during meiosis 1, during prophase 1 of meiosis 1, there is something known as crossing over. What is crossing over? Crossing over is exchange of genetic material between two chromosomes. One can be paternal, one can be maternal. So, exchange of genetic material between two chromosomes is known as crossing over. Okay, so because of exchange of genetic materials between two homologous chromosomes, there is genetic variability which arises in the cell which is forming after telophase 1 of meiosis 1. Okay, so why is genetic variability occurring in all the offsprings of the meiosis? Because of the crossing over, that is exchange of genetic material that occurs in the prophase 1. Okay, I hope this is clear to you. So what are the options saying? Let us say the options are saying cytokinesis, replication of DNA during S phase. Cytokinesis is division of cytoplasm. Replication of DNA during S phase is not the reason for genetic variability among offsprings of meiosis. Okay, spindle formation. What is spindle? Mitotic spindle is a protein which helps bring chromosomes near the equator of the cell. So that is definitely not the reason why there is a genetic variability between offsprings of meiosis. Okay, so this is also wrong option. So first 1, 2, 3 are ruled out. So what option we are left with? We are left with fourth option. So the answer to 101 question is option number 4. I hope that was clear to you. If it is not, then replay the video and you will understand. Okay, so before I move on to the next question, I also want to say that during packaging stage of prophase 1, this crossing over occurs. Okay, so I hope this is clear to all of you because it is a very easy question. The first question is very easy. I cannot even emphasize how easy this is. It's an extremely easy question. I would say this is not even NEET level at this point. This is so easy. So moving on to question 102. Let us see what it is saying. Okay, so this is a fill in the blank type question. You have to fill in the blanks. So this is very easy. I think this is plain memory. If you remember, you remember. If you do not remember, you have to just go to the NCERT and read. So fill in the blanks. Light saturation occurs at what percent? At 10% of full sunlight. When a plant is receiving 10% of the full sunlight, there is saturation of light, which means plant cannot further utilize light energy. So at 10% of the maximum sunlight, that 10% of the maximum sunlight when reaches the plant, that is when the plant is fully saturated and cannot receive more sunlight, or to be precise, cannot utilize more sunlight. Okay, now the second option, what does it say? It is say there is a dash relationship between incident of light and carbon dioxide, incident of light and carbon dioxide fixation rates at low light intensities. So what does it mean? It means that when there is low light intensity, when there is not so much sun, when it's very little cloudy, little sunny, at those intensities of light, the carbon dioxide fixation is directly proportional to the amount of light that the plant is receiving. So if plant is receiving, let's suppose 1% light, then incident light, then the rate of carbon dioxide fixation also will be 1%. That is, the rate of photosynthesis also will be 1%. So there is a linear relationship between incident light and carbon dioxide fixation at the low light intensities. Okay, I hope that is clear. So this is linear here, which means it is directly proportional. Okay, now what is third option saying? It is saying the C3 plants show carbon dioxide saturation. This is plain memory at what? 450 microliter per light. Okay. And while C4 plants show at what? 360. So the saturation to light is higher for C3 plants and the saturation to light is low for C4 plants. Okay, so this is direct your NCRT bit. This is plain memory. You do not have to use your brain or there's no concept. This is plain memory. Okay, so you have to read NCRT. There's nothing you can do. Now moving on to question number 3. So yeah, so I did not say the answer to question 103. 1 second. The answer to question 1 second. Yeah. So the answer to question 102 was, the answer to question 102 is, 1 second guys, is option number 4. Okay, I hope it is clear. Okay, I'm sorry, some technical glitches. I'm not a very technology friendly person. Okay, so moving on to question 103. What it is saying, let's choose the correct statement in the process of protein synthesis. Okay, okay. After uncoiling of DNA molecule, one strand acts as template for the formation of mRNA. Yes, this is true. One is template strand, which is used for forming mRNA, and other is coding strand. Right? So this is true. This is not wrong anywhere. Now let's see. What is the second option saying? It is saying, in the presence of RNA polymerase enzyme, the mRNA is formed based on triplet codes. Protein is formed based on triplet codes. mRNA is never formed based on triplet codes. mRNA is formed with concept of complementarity. So the template strand of the DNA is the main strand, and the nucleotides of the mRNA are complementary to the template strand of the parent DNA. Okay, so the DNA from which we are forming this protein, the templates, there is a template strand, and complementary to the nucleotides of these template strand, there is an mRNA which is formed. Okay, that is correct. But mRNA is not formed based on triplet codes. Based on triplet codes, what is formed? Protein is formed. So this is wrong. Okay. So translation occurs based on triplet codes. So let's suppose this is MR, which has codes like AUC, CAA. So based on the 3-3 triplet code, the amino acid will be decided and it will be added on. So this occurs during translation. It does not occur during transcription. Okay. Transcription is synthesis of mRNA from DNA, and translation is synthesis of protein from mRNA. So during transcription, what happens is the DNA uncoils and there is a template strand based on which there is new mRNA formed. Then the codes, nucleotides in the mRNA are complementary to the nucleotides present in mRNA. Okay, and during translation, what occurs? What happens is, based upon the triplet codes of the mRNA, amino acids are bought by tRNA and they are added on each other through your protein synthase, and then a new protein is formed. Okay, so this is a wrong statement. Statement number 2. Third statement, the mRNA that leaves nucleus reaches cytoplasm and gets attached with 30ase ribosomal unit. Okay. mRNA that leaves nucleus reaches cytoplasm and gets attached with not 30S. It does not get attached with 30S ribosomal unit. This is wrong statement. The amino acids are transferred from one intracellular. The amino acids are transferred from the intracellular amino acid pool to the active ribosome by mRNA. No, they are transferred by tRNA. So this is also wrong statement. This is also wrong statement because it does not get attached to 30S RNA ribosomal unit. Okay, so I hope this is clear to you. So if the statement 2 is wrong, statement 3 is wrong, statement 4 is wrong, then which statement by default is correct? The statement which is correct is, yes, statement number 1. Which statement is correct? Statement number 1. Okay. So the correct statement is, correct statement is statement number 1. Okay, so answer to 103 is 1. Okay, so let us see 104 question. Yes. So what does 104 say? It says select the correct sequence of steps in DNA fingerprinting involving southern blotting hybridization using radio label BNTRS probe. Yes. So they basically want us to select the southern blotting technique, steps of southern blotting technique in sequence, starting from the first step, ending with the last. Okay, so what is the first step? Let us see. Let's read first all of the steps and then we will organize them based on the sequence, starting from the first one and ending with the last one. So the first step is hybridization using labeled VNTR probe. I mean, the first option is hybridization using labeled VNTR probe. Okay. The second is isolation of DNA. Third one is transferring of of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, detection of hybridization DNA fragments by autoradiography. What is autoradiography? Nothing. They just radio labeled particle is attached to the DNA fragment which is to be detected, and then they see it under UV light, and that radio labeled particle which is attached to the DNA to be detected shines. That's how you identify that this is the DNA to be detected. Okay. So separation of DNA fragments by electrophoresis, digestion of DNA by restriction endonucleases. So southern blotting, nothing but they just want us to arrange the steps of DNA fingerprinting in sequence. You do not have to be very, oh my God, what is southern blotting? What is southern blotting? No, you should not be. Southern blotting is a technique of DNA fingerprinting. That's what you should know. So these are basically the steps of DNA fingerprinting, which you should arrange it in order, starting from the first one and to the last one. So what is the first step in this? The first step will be isolation of DNA. Second option. Then the second step would be digestion of DNA by restriction enzyme. After digestion of DNA, what do? We separate the fragments of the DNA. So third step would be separation of DNA by electrophoresis. Then after that, what do? We move those fragments, separated DNA fragments to nitrocellulose. So fourth step would be third option. Okay. After moving the fragments of DNA, what do? We hybridize them using the NTR probe, which means we select the probe DNA and put that probe DNA on the isolated fragment DNA. Okay. So that is hybridization. We select a probe DNA and put that probe DNA onto the DNA fragments which we have already separated using electrophoresis. So that is a fifth step, which is option number 1. Then after we have put the VNTR probe to the separated DNA fragments, we now check using autoradiography, which is sixth one step. So the step would be something like 2, 6, 5, 3, 1, 4. 2, 6, 5, 3, 1, 4. So answer is second option. Now, let me tell you something. What is VNTR probe? Nothing. What is VNTR probe means variable number of tandem repeat probe. Okay, variable number tandem repeat. What is this probe? This probe is nothing. What you should do, what is this? It's a basically a small fragment or small DNA segment of DNA which has a repeats of nucleotides. So let's suppose if it is like, let's say CAA, then CAA, then AUC, then again CAA, then again AUC. So it's a small segment of DNA in which a sequence of nucleotide is repeated at different numbers. So what is that? It is variable number of tandem repeats, a fragment of DNA which is having sequence of nucleotides which are repeated in different numbers. That is variable number of tandem repeat probe. Now, this probe is labeled with a radioactive substance and it is put to the DNA fragments, okay, which we have separated. Now, this probe will go and bind to the DNA fragment which is complementary to it, and it will make it radioactive. And when we put UV light during autoradiography, the DNA fragment which this VNTR probe binds to, it shines in the UV light, making us identifying that complementary DNA to the VNTR probe. Okay. So this is what DNA fingerprinting is about. So answer to this question is second option. Now moving on to fifth option. Fifth question, what is it saying? Match the two of them. This is a very easy question, if you ask me. One second. Yes. So this is the most easiest question I have ever read till date. What is a chlamydomonas? It is an algae. So chlamydomonas is an algae. Cycas is a gymnosperm. Selaginella is a pteridophyte. Sphagnum is a moss. So answer is A3, B4, C2, D1. A3, B4, C2, D1. A3, B4, C2, D1. So correct option is second option. Okay. Moving. Yes. So moving on to question 107. Let me 1 second. We'll see. Yes, 107. Give the correct descending order of organism with reference to their estimated number found in Amazon forest. So my friends, this is very, very, very memory-based question. You should not remember anything. This is like total memory. Read NCERT, remember and put it on, vomit it on your paper. So this is what it is. So in Amazon forest, more than 40,000 plant species are found. More than 1,25,000 invertebrates are found. Then more than 3,000 fishes are found, 427 mammals, and your NCERT states that there are 1,300 birds. So we have to arrange it in the order in descending order. Okay. So descending order is largest to smallest. So the biggest one is P, invertebrates. After invertebrates, we have plants, A. After plants, we have fishes. After fishes, we have birds and then mammals. So it is B, A, C, E, and D. So answer would be fourth option. Okay. Fourth option is the correct answer to the question. 107. 7. Okay, 1 second. Okay. So this is a very easy question, completely fact-based. And question 108. What is question 108 saying? It is saying, it is a fill in the blanks type question. You have to just fill in the blank. Spraying sugarcane crop with what? With dash increases the length of the stem, thus increasing the yield as much as dash tons per acre. This is direct from NCERT. So it is, if you spray the sugarcane crop with gibberellin, it will increase yield of gibberellin, yield of sugarcane by 20 tons per acre. Okay. Then The second statement is dash does not occur naturally in plants. Kinetin. I'm sorry, one second. One second. Kinetin does not occur naturally in plant. Okay, kinetin does not occur naturally in. Search for natural substances with cytokinin-like activities led to isolation of dash from the corn kernels and coconut meal, lead to isolation of zeatin. Okay, so answer is fourth option, A, B, C, D. A is gibberellin, B is 20, C is kinetin, and D is zeat. So this is also directly from NCERT. This is just like factual memory based. There is no concept here. You just have to read NCERT, remember, and vomit in paper. That's it. Okay, so let us move on to the next 109 question. It says, identify the type of plant tissue from the properties given below. So this is from your anatomy of flowering plants chapter. Very easy, very easy. Let us read. So first characteristic is forms major component within organs. Second one is cell wall thin and cellulosic. Okay. Shape of cell is isodiametric. Okay. What type of cells are isodiametric? Parenchyma is isodiametric. Yes, this is what parenchyma has thin cell wall, which is cellulosic. Yes. It forms major component. True. It is a part of cortex. Yes, true. Intercellular spaces might be present or absent. They are photosynthetic. They help in storage and they have secretory function also. All these are features of, yes, these are features of parenchyma. So parenchyma has a thin cell wall, which is made up of cellulose. In this, intercellular spaces are mostly absent, but might be present also. The functions of parenchyma as listed by NCERT are photosynthesis, storage, and secretion. And it is the major part of plant body because it is mostly involved in formation of cortex. Then moving on to question 1010, what does it say? The general egg apparatus of the embryo second angiosperm consists of one egg cell and two synovials. This is so easy, so easy. Like I cannot tell you that this is the easiest question possible from your sexual reproduction in flowering plants. Moving on to question 111. This is also purely memory based and I love this question. This is so good question. So it says match list 1 with list 2. 1 second. Match list 1 with list 2. So imbricate type of imbricate is 1 second. Yes, imbricate type of estivation is seen in Cassia. So imbricate estivation is seen in Cassia. Valvate estivation is seen in Calotropis, very famous. Waxillary estivation is not seen in P, it is seen in B. But in cotton, what do we see? We see twisted estivation. So that would be, that is D. And in B, which is a Fabaceae family, we see what? What do we see? Our axillary estivation. So that is C. So answer would be A1, sorry, A2, B1, A2, B1, C4, D3, A2, B1, C4, D3. Second option is the correct answer to 111 question. Moving on to 112 question. The phenomenon by which undividing parenchyma cells start to divide mitotically during plant tissue culture is called as, okay, so they were not dividing first, which means they were differentiated and then they started again to divide, and this is called as de-differentiation. What it is called as? De-differentiation. Okay, so answer is second option. I hope that is clear to you. Moving on to 113. This is also extremely easy question. In meiosis, crossing over and exchange of genetic material between homologous chromosomes, as the first question was saying, are catalyzed by the enzyme. Ooh, interesting. It is not asking is seen in which phase of prophase, but it is saying is catalyzed by which enzyme. Yes, so this is catalyzed by recombinase enzyme. It is catalyzed by recombinase enzyme. This is little out of NCERT, but you should remember It's a very easy question. If you do not know, note it down. Then moving on to question 114. 114 says, what is the 5-carbon compound which is formed in Krebs cycle? So Krebs cycle, we have alpha ketoglutarate acid, which is a 5-carbon compound. Okay. We have alpha ketoglutarate acid, which is a 5-carbon compound. Okay. The answer is first option. This is very easy. Okay, so alpha ketoglutarate acid is a 5-carbon compound. There's nothing called oxalosuccinic acid. It is oxaloacetic acid. Blunder this is actually. This is a 4-carbon compound and this is also a 4-carbon compound. Okay. The chromosome theory of inheritance was proposed by who? This is also very easy to proposed by Sutton and Bovary. Thomas Morgan, what did he do? Recombination. Mendel worked on pea plant. Okay, Robert Brown is related to nucleus. Okay, so all these is different scientists with different achievements, which you should know. I hope this is clear to all of you. Moving on to the question 116. This is a large question. Let us read what the question wants us to do first. Read the following statements and find out the correct ones. Okay, very easy, very easy. Yes. So the first statement says, in plant growth and differentiation, both are open. Yeah. Okay. They keep on growing and both are open. This is a correct statement. Okay. I am so used to putting a cross that for a correct statement also I am putting a cross. The final structure of a cell or a tissue at maturity is also determined by the location of the cell within the plant. Yes, true, true. True. Final structure of tissue is decided by where the cell is located. The final function of the cell is decided where the tissue or the cell is located in the plant. Let's suppose if it is present in photosynthesis, it will be if it is present in leaf, then function would be photosynthesis. If it is present in root, then function would be absorption of water and minerals. So depending on the location of the cell in the plant, its function and structure is decided. This is true. Cells positioned away from the root apical meristem differentiate as root-capped cells, while those pushed into the periphery mature as epidermis. This is also true. This is true. Environment signals such as light and gravity affect certain stages of growth. Yes, yes, they do. Intrinsic factors include both intracellular and intercellular factors. Okay, while extrinsic factors include light, temperature, water, oxygen, and nutrition. These all statements are true. So answer to this question is all of the above. This is directly NCRT taken question. Okay, so if you have read NCRT, this would be a cake for you. Cakewalk for you, not cupcake. I'm sorry, cakewalk for you. It is a very easy question. Okay. Directly taken from NCRT. Whenever you see a long question, just remember that the question is definitely going to be easy because half of the people are just going to see the question and feel like, oh my God, this is so long, and they are not going to attempt only. That is where you should not leave the question and you should attempt. Okay. This is now another extremely easy question. Read biodiversity. This is what you have to read maximum. What is that? This is this only. Okay, conservation. XC2 in situ conservation. So what are sacred grooves? Sacred grooves? Sacred grooves, they are found in Khasi Hill. I call it Khansi Hill, but I don't know, you can call Khasi Hills also. So this is A. Then zoological park. Zoological park is Zoological parks is ex situ conservation. We do zoological park in zoological parks. Animals are not protected in their natural habitat. They're protected out of their natural habitat. So this is ex situ conservation. Then Nile perch is an alien species. Alien invasion leading to biodiversity loss. Okay, Amazon forest releases large amount of oxygen because it has large amount of species of plant. Okay, so it is A4, B3, C1, D2. A4, B3, C1, D2. So answer is first option. Okay, I hope that is clear to all of you. Then moving on to question 118. What does it say? It says bacteria. You have to match the number of base pairs of nucleotides basically. So bacteriophage 174 has, this is plain memory, 5,386 nucleotides. Bacteriophage lambda has 4,8508 nucleotides. I remember the first number of it. That's how I remember. You can also try that one. E. coli has what? 4.4, 4.6. This I remember. 4.6 into 10 power 6. That is not 106, that is 10 power 6. And then this is haploid human DNA, 3.3 into 10 power 9 base pairs. So this is C. Oh, I'm sorry, this is D. So A2, B1, C4, D3. A2, B1, A2, B1, C4, D3. So correct answer is third option. Okay. Okay. Ribosome, given our two statements, we have to choose the correct option from the following after reading the two statements. So the first statement says ribosomes involved in protein synthesis. True. Ribosomes are involved in protein synthesis. Ribosomes are membrane-bound organs. Now this is something very wrong. Ribosomes are non-membrane bound organs. Okay, they are not membrane bound. So correct option is option. Statement 1 is correct. Statement 2 is incorrect. So option 2 is the correct option to this, which was okay. I hope again now this is clear to all of you. Moving on to the question 120. What does it say? In nature, in inter, in nature, in interspecific competition, they point out that species facing competition might evolve mechanisms that promote coexistence rather than exclusion. One such mechanism is resource partitioning. Resource partitioning is division of resources in such a way that the resources which are used by one species will not be used by other species to avoid competition between two different species so that both of the species can coexist in that environment. Okay, this is a very easy, extremely easy question. I would rate it 1 by 10 on the level of difficulty. Okay, so this is your question 120. Answer is second option. Okay. I hope this is clear to you. Yes, so now let us move on to question 121. Let's see what is it saying. So the question 121 is saying Golgi body is associated with which of the following? What is Golgi body? Golgi body means it is referring to Golgi apparatus. Golgi apparatus is associated with which of the following is the question. The Golgi apparatus is associated with secretion of different substances. Yes, it is true. Different substances like glycoproteins, glycolipids are secreted from Golgi apparatus because glycoprotein and glycoproteins are synthesized. They are synthesized inside the Golgi apparatus, and after synthesis, they are secreted from them. And Golgi apparatus also helps in packaging and storage of different materials. So all of these are functions of Golgi apparatus, which was found by Camille Golgi or Golgi, whichever way you want to say it. So the answer to question 121 is answer number 4, option number 4. Let us see 122. So the 122 is asking that the grana which are present, grana means stacks of thylakoid which are present inside the chloroplast, they are connected by which of the following structures? So grana which are present, that is stacks of thylakoid which are present inside the chloroplast, are connected by stroma lamella. Stroma lamella are present in stroma, of course. So answer to the question 122 is option number 3. I hope this is clear to you. Moving on to question 123. This is also very simple fact-based question. You do not have to, conceptualize anything in this. So the question is something like, one second. Yes. So the question is something like, in electron transport chain, the ETS, in electron transport chain, what is complex 5? Complex 5 in electron transport chain is referred to as what? It is referred to as ATP synthase, also known as ATPase. What it is referred to as ATP synthase, also known as ATPase. So answer to this question is option number 2, or ATPase. Okay. ATP synthase helps in synthesis of ATP, that is adenosine triphosphate from ADP plus inorganic phosphate. Okay, let us see question 124. What is it? What it is saying? It is saying there are two statements. The first statement says Mendel crossed true breeding P lines. Yes, this is correct. What is true breeding P lines? True breeding P lines means homozygous pea plants. Yes, that is true. Mendel did cross homozygous pea plants. Okay. With any characteristic, either height or pot color, seed color, any characteristic, Mendel crossed homozygous pea plants. The true breeding pea lines, that is homozygous pea plants, they show stable trait inheritance. Yes, that is true. They do show stable trait inheritance and expression of several generations during selfing. It means they show stable trait inheritance means it means that the inheritance of their characteristics, characters of their characteristics or inheritance of the traits in a cross between two homozygous pea plants is stable throughout the generation. It does not vary much. It does vary, but it does not vary much. It's stable throughout the generation. If there is a cross between two homozygous plants, and if that cross is continued, then the new generations that are formed are stable, which means they do not vary a lot from the parent plant. They do vary, but do not vary a lot from the parent and from their colleagues a lot. Okay, so that is what is this statement 2 saying. So statement 1 and statement 2 are correct. So answer would be both statement 1 and 2 are correct. Answer is fourth option. Moving on to question 125. Oh my God, this is the easiest question existing on planet. It is saying which of the following is powerhouse of cell. Now, if you do not know this since class 8, I don't know why you should know. Okay. So next question is question 126. What is it saying? Let us see. What is question 126 saying? It is saying which one of the following experiment? It is saying in the following experiment, which okay. It is saying which among the following experiments have used radioactive substances for their experiment. So answer would be, let's see. Griffith did not use any radioactive material for his experiment. Hershey and Chase, yes, they used radioactive sulfur and radioactive phosphorus for their experiment. Okay. Michelson and Stahl. Okay, let's see. Taylor also used radioactive thymidine for his experiments. Morgan used housefly. He did not use anything for his experiment. And what is Michelson and Stahl, they used nitrogen 15 for their experiment, and nitrogen 15, my friends, is heavier than nitrogen 14, but it is not radioactive. So the question option 3 also goes to wrong option. So answer would be option B and option D, which is second option. Question 126. Predict the following chart. Okay, so they want us to predict the disease, characteristic of the disease which is being caused in this chart. Okay, so here the female is sick, right? The male is normal. So female is sick but not affected. So this is sick but not affected. That is called carrier. Male is normal. From carrier female, it is passing on to male, not male. So here this male is diseased, not diseased, diseased. Female is normal. Another female is also normal. Another male is diseased. It means that here the female is a carrier. Okay, and from female to pass from another female, here the female is a carrier. Okay, if it is a dominant disease, it will pass on to female also. It will pass on to male also, right? Because it is a dominant disease. One X chromosome is sufficient for disease to be caused. But that is not the case. It is being passed only to the male. It means it is not dominant. So it is a recessive disease. Okay. Now, this female is a carrier. It is passing only to the male. It is passing to male, which is only to males, not to females, which means it is a recessive disease. Okay, it is not dominant because if it was dominant, it would have been caused in females also. Now, because it is because it is recessive and caused only in males, it is X-linked. Because if it is because if it was autosomal, then it would not have been caused in males also. Okay. So let us see, I'll tell you how. So let's suppose if it is color blindness only. Let's see. So this female is X. Okay, 1 second. So this female is not color blind, but she's affected. So XCX, which means she is affected but not color blind. Male is normal, means he has XY. Okay. Now, if we cross these two people, there can be one possible genotype of XY and one possible genotype of XCY and two possible genotypes of XCX, XX. Okay. Or this can be XX also. Depends. It can be XCX also, XX also, or it can be, this also can be XCX or XX also. Okay, or both can be XX or both can be XCXC, XCX, XCS. Okay, now let us see. So, see, this is XXI. Okay. So now see, this clearly is XCY, means this male is affected. Okay. Now this affected male is marrying a normal female, which is XX. She's not a carrier. So the possible would be XX, okay, XY, XCX, right? See, one female is a carrier. So this disease is a X-linked recessive disease. X-linked recessive disease. Example for X-linked recessive disease is color blindness and yes, and hemophilia. So answer would be character is recessive carried by X chromosomes. Character is recessive, codominant. No, character is recessive, but it is not codominant. That is a wrong question, wrong option. Okay, character is recessive, but it is not codominant. So fourth option is not the correct answer. Character is not dominant. So first option is also not correct answer. Character is carried by Y chromosomes. No, that is also not the correct answer. If character was carried by Y chromosomes, female cannot be a carrier only because females absolutely do not have Y chromosomes. Then character is sex-linked recessive. Yes. Third option is correct answer. Moving on to question 128. What does it say? Which of the representative figures showing an individual inflicted with? Okay, so this is a classical figure of an individual suffering from Turner syndrome. Individuals suffering from Turner syndrome would have a genotype of what? Would have a genotype of 44, 45 plus XO, which means they have one less X chromosome. And only females are affected. Only females affect from are affected from Turner syndrome. They have irregular no menstrual cycles. They have no menstrual cycles, rudimentary ovaries, etc. Okay. So the answer to question 128 is option number 2, which is Turner syndrome, which is caused by absence of one X chromosome. Having people of Turner syndrome have genotype of 45 plus XO. Okay. They cannot reproduce. They have rudimentary ovaries, small breasts, male pattern hair, etc. Okay. 129. The number of documented basmati rice varieties grown in India are how many? 27. Okay. Now, 130. Given below are two statements. Statement one, population size, technically called population density. Size is technically called population density. What is statement two? Mortality is number of deaths in population during a given period of time. This is wrong. Okay, this is mortality is number of deaths in a population during a given period of time. Okay, this is correct. Okay, this is correct. Mortality is number of deaths in a population during given period of time, and morbidity is number of diseased people in a population at a given point of time. So it is correct. Population size is basically population density, which means how many people are present per unit area at a particular given point of time. So statement one and statement two both are correct. So answer to this question would be option number 4. Answer to 130 is option 4. Moving on to question 131. If a new habitat is just being colonized, which process may contribute more significantly to population growth? Okay, immigration. A newly colonized habitat requires people to come in there and live and then reproduce to produce new children. Okay, you cannot, if you need people to reproduce first, reproduction does not happen without people. So first we need people. And how will people come without reproduction? First, through immigration. People move into that area and then reproduce. So immigration is required to colonize a newly developed area. Okay. So that is question 131. Option 2 is the correct answer. Moving on to question 132. What does it say? Which of the following is not entirely a true fact? Competition occurs when closely related species compete for same resources that are limiting. Competition occurs when closely related species compete for same resources that are limiting. Okay, that is true. That is true. When there is a resource which is limiting and two species require that resource, they will fight with each other for that resource, and that is what is called as competition. True statement. Okay. Totally unrelated species compete for same resource. Totally unrelated species compete for could compete for same resource. Second. Yeah, exactly. Now, if let's suppose human being wants chicken to eat, lion also wants chicken to eat, we both would, and there is only one chicken for eating. We both would compete. We are totally unrelated species, but we can compete with each other. That is a true statement. Okay. True statement. Resources need not be limiting for competition to occur. Interference competition, the feeding efficiency of yes, might be reduced to interfering and inhibitory presence of other species. Yes, even if resources are plenty. Yes, that is true. There is no complete need of resource to be limited for competition to occur. Okay, competition can occur in absence of limiting resources also. And what happens in such cases? What happens in such cases? One species interferes with feeding capabilities of another species, and this gives inhibitory effect to the feeding capabilities of other species. Okay, even if the resources are plenty for both of them. Fourth option, what does it say? Complex interplay of interspecific interactions and environmental variability characterizes community structure. This is direct NCRT statement. I need not even explain. It is saying that there is a interaction between environment and interspecific interaction, and that leads to how a community is formed. So community structure is formed based on how species are interacting between each other and what is the environment variation. Based on that, communities are formed. That is what is option 4 saying. Okay. So now let us see, we have to find what not entirely true. So we have to find a relatively false statement. So the competition occurs when closely related species compete for same resources that are limited. Competition does not always occur when closely related species are competing. Very far related species can also compete for the same resource. That is what the second option saying. So this statement is not entirely correct. This is correct, but not entirely correct. A true fact. Okay, so answer is first option. These all are direct NCRT lines. Now question 133. So here we have assertion and reason type of question. Here they are asking assertion, Cuscuta is a parasite. Yes, it is true. Cuscuta derives its nutrition from the host plant. True. Which it parasite is. Yes. So why is Cuscuta a parasite? Because it derives nutrition from the host plant, which parasitizes. Parasitizes. Okay. So answer would be both A and R are true, and R is the correct explanation to A, which is fourth option. Okay. Moving on to 134. Examine the food web organism in ninth position. Okay. So this is a herbivore. Okay. So ninth organism basically is eating a herbivore and a carnivore, which means it's an omnivore. So answer would be fourth option. Moving on. Question 135. It says when a flower which can be divided into two equal vertical halves only in one particular plane is called as radio zygomorphic. When a flower can be divided into two equal halves only in one particular plane, it is zygomorphic. Zygomorph. Section B, 136. Apocarpus condition is apocarpus means fused. Apocarpus means fused carpels. So fused carpels are found in which of the following? They are found in lotus and rose plant, both flowers. Okay, they are found in lotus and rose. Ovary is half inferior in flowers of. Ovary is half inferior in flowers of. Yes, plum. In plum flower, there is half inferior ovary. Moving on to keel is a characteristic of flower of Fabaceae family, which is B. First option is the correct answer. This is such an easy paper. I love this paper. Select the correct match. Dicot stem. Okay. Sclerenchymetus hypodermis. No, wrong. Monocot stem. Parenchymatous hypodermis. True. Dicot leaf parenchymatous mesophyll is made up of parenchyma and dicot leaf. Okay, true. Monocot leaf, we have bulliform stem. True, we do not have parenchymatous hypodermis in monocot stem. Okay, we do not have parenchymatous hypodermis in monocot stem. Hypodermis is what? Hypodermis is sclerenchymatous in monocot stem and cholenchymatous in dicot stem. Cholenchyma and this is sclerenchyma. So answer is 3 and 4. 3 and 4, fourth option. Okay. 140. Which of the following is not included in stomatal apparatus? Sclerenchyma cells are not included in stomatal apparatus. Oh my god, this paper is extremely easy. Open vascular bundles are found in. Come on, open vascular bundles are found in. Yes, they're found in dicot stem. Because stems undergo secondary growth. And what are open vascular bundles? Open vascular bundles are bundles which have cambium in between them. Cambium present between phloem and xylem. Okay. After evaporation of all the water from living tissue, we get, yes, after all the water from the living tissue is evaporated, what do we get after evaporation of all the water from the living tissue? One second, one second. So, after evaporation of all the water from living tissue, we get dry weight of the living tissue. Now, which of the following is a nucleoside? Okay, nucleoside is thymidine, cytidine, all are uridine. Okay. Adenosine and guanoside are nucleosides. Uridine is a nucleoside. Thymidine and cytidine are also nucleosides. So, all of them are nucleosides. Nucleoside is made up of sugar plus nitrogen base. Okay, if you add phosphate to this, it will be a 9-nucleotide. Recombination is completed by which stage in the meiosis? It basically wants to ask you that after which stage you can say that recombination or that is crossing over is finished. So after packaging, the crossing over is finished. So answer to question 144 is option number 3. The characteristic feature of diplotin stage, one second. The characteristic feature of diplotin stage of meiosis 1 is recognized by diplotin stage is X-shaped structure. Chiasmata are formed in diplotin. X-shaped structures called chiasmata are formed in diplotin stage. Okay. Okay. Dissolution of synaptonemal complex also occurs. Okay. Okay. Homologous chromosomes of bivalent start to separate with each other. All of these features are seen in diplotin stage. So answer to this question would be fourth option, that is all of the following. 146. Branch of botany which deals with internal organization in plants is known as anatomy. 147. Chromosomes are moved to spindle equator in which phase? In metaphase. Chromosomes are moved to equator in metaphase of meiosis 1 or 2. Okay, always in metaphase. Centromere splits in anaphase 2. In anaphase 1, centromere does not split. Remember, in anaphase 1, centromere does not split because two homologous chromosomes separate. Pairing of homologous chromosomes occur in zygote. Crossing over occurs in packaging. So A1, sorry, A2, B1, C4, D3. First option is the correct answer. Morphology of chromosome is easily studied in which stage of cell cycle? Morphology of chromosome is easily studied in, yes, metaphase of cell cycle. Which is the first terrestrial plant which possesses vascular tissues? Bryophytes or pteridophytes? Pteridophytes are the first plants to have vascular tissues. Read the following statements and find out the incorrect statement. We have to find out. Okay. Embryo development precedes endosperm. True. Embryo is formed before endosperm is formed. Though the seeds differ greatly, the early stage of embryo, though, wait a second, wait a second. Embryo is formed before, precedes means before. Okay, I'm sorry, this statement is wrong. Embryo is formed after the formation of endosperm. Endosperm is formed before embryo formation. Endosperm is formed at the time of zygote formation only. Okay, whereas first zygote forms, then embryo forms. So there's a time between formation of endosperm and formation of embryo. First the zygote will be formed, then embryo will be formed, and immediately at the time of simultaneously with zygote formation, the endosperm also forms. So endosperm forms before embryo forms. Okay, and it provides the embryo nourishment. So this statement is wrong. Though the seeds differ greatly, early stage of embryo development are similar in both monocots and dicots. Yeah, early stage is what? You form zygote, then cleavage, and then all those divisions occur. So early stage of embryogenesis is same. Okay, even though the seeds of monocots and dicots are different extensively, but the early stages of embryo formation are same. So this is true statement. A typical dicot embryo consists of embryonal axis and two cotyledons. That is why it is a dicot embryo, right? So this is also true. And what is it? Just endosperm may either be completely consumed in a developing embryo. Example: castor and coconut have embryo. They are not embryo lack. So this is wrong statement. They have embryo. Sorry, they have endosperm. When endosperm is completely consumed, they are non-endospermic, right? So example for that would be pea, maize, like that. Whereas castor and coconut, they are endospermic. They have endosperm. Okay. So that this statement is wrong. Coconut water from tender coconut is, no, it is free cellular, free endo. Coconut water is a free endosperm or otherwise known as nuclear endosperm, and the kernel, white color kernel, is cellular endosperm. So here this is also wrong. So we have D, E, and the A statement is wrong. So answer would be A, D, E. Fourth option is the correct answer to question 150. Okay, this paper was extremely easy. I cannot emphasize how this paper was. And if you have any doubts, please feel free to put them in comments. And if you feel that I have done any mistake while teaching you, feel free to correct me also. And yeah, before moving on further, I will clarify something about question 106. Let us see where is 106 question. Yes. One second. Okay, let's start. I'll just clear all the mess I found. I told you, I think that we will discuss about 106 question in the end, and let us see then. Or I might have skipped it for some reasons. Yeah. So moving on to question 106. It is saying a segment of DNA has triplet base sequence of A, C, G, A, C, A, G, C, C, G, C, A, C, A. Okay, this is the triplet base sequence, and due to mutation, this base pair, it got deleted. So what would be the effect of it on the DNA is what the question asking. So there will be complete change in the type and sequence of nucleotides. Yes, that is true. Initially, AAC, GAC, AGC, let's suppose, cited in thymidine, cited in thymine. I mean, if we talk in terms of nitrogen bases, let's suppose it is cytosine, thymine, uracil. All these are the nitrogen bases which are formed, right? All these are the nitrogen bases which are there according to the triplet or thymidine, cytidine, uridine. All these are the nucleotides. But due to this change in first nitrogen base, now the sequence would be like ACG, ACA, GCC, GCA. One second. Yes. GCA, ACG, ACA, GCG, GCC, GCA, CAA, AA. The sequence would be like this, right? So amino acids would be formed according to this sequence, which is totally different from what? From this sequence. So the amino acid formed in this chain would be totally different from what that would have been formed in this sequence. So this is correct. This is the effect that will happen. Polypeptide will have one amino acid less. No. No, for one amino acid to be less, this totally three base pairs should be deleted out of the codon. One deletion of one base pair will not lead to deletion of complete amino acid. There will be no change in poly. No, there will be change. There will be change. The sequence of amino acids in the proteins formed will be changed. The first amino acid will be different only. So here only is written. The question is the only first amino acid will be different. No, that is also not true. All the amino acids in the complete chain of protein will be different if first base pair is deleted. Okay, so this is also wrong statement. So answer is first option. So answer to question 106 is first option. Okay, so I hope 106 is also clear to you. And this is what is FLT 1 botany paper about. It is extremely easy. I cannot find any hard question in this question paper. And if you have doubts, if you feel that there is some here and there, then you can always put it up in comments. I will be happy to reply. And all the best for your preparation for NEET. And see you in next video. Bye.